now invite Professor Hartl for the second part of the lecture. The title will be Cellular Machineries of Protein Folding and the Implications in Human Disease.
Yeah, thank you very much. I would like to start by saying what a wonderful time we are having here in Hong Kong, and thank all our hosts for the warm hospitality and really great excitement that we are experiencing. Obviously, the Shaw Prize and the whole ceremony was really quite amazing. Now, I have to say at the outset that there will probably be a bit of overlap with what you just heard from Art. That is unavoidable given the close connections of his and our work, and of course our initial collaboration in the early phase of discovering the chaperonin machine. Now, you all know that protein molecules are probably the most important macromolecules in cells. They are responsible for almost all biological functions. And just like the famous terracotta soldiers, each of the approximately 10,000 proteins that we have in our cells has a different face. It has a different shape, a different fold, and that's required for it to carry out its particular biological function, even though, of course, the general building plan is very similar. Proteins are synthesized as chains of amino acids on ribosomes. You just heard about all this, and in order for them to become functionally active, the extended polypeptide chain has to collapse into a highly defined three-dimensional structure, and this is the process that we call folding. So folding is clearly then one of the most important processes in biology, because it's the final step for the transfer of information from the gene to the functional protein molecule that then carries out the particular biological effect. As you heard from Art, 50 years or so ago, Christian Anfinsen did pioneering experiments in which we could demonstrate that at least certain small proteins could refold spontaneously from their unfolded states when they were artificially denatured. And these were, of course, highly influential experiments that suggested strongly, and in fact did so for many years, that the protein folding process is generally a spontaneous reaction, even though it became clear relatively soon that the classical Anfinsen experiment doesn't actually work for many proteins, particularly when you go to larger proteins with more complex structures. Most scientists did not really consider the problem of protein folding as something that should be addressed from a cellular viewpoint. Now, this has obviously changed over the last 10, 15 years. We now know that the initial folding of proteins, the de novo folding of proteins, at least for many molecules, probably the majority of proteins in the cell, requires the machinery of molecular chaperones, as I said, for de novo folding, but also, as we are beginning to appreciate more and more in recent years, this machinery is required for the maintenance of the proteome in a functional state, a state that we would define as proteostasis. This maintenance of the proteome requires also molecular chaperones, particularly then under conditions of conformational stresses such as heat stress or other forms of stress, where pre-existent folded proteins begin to lose their folded structures and then require the molecular chaperones to refold or go through other mechanisms of protein quality control. Now, we were very fortunate to be able to contribute to this change in paradigm, this new view of how proteins fold in the cell through experiments using the mitochondria. You just heard from Art about these experiments, exciting phase of collaboration that we had. I was at the time a young scientist and leader of a small group of students in the department of Walter Neupert at Munich University. And Walter Neupert is one of the, or at that time was one of the two main experts in studying how proteins are sorted within the cell from the cytosol into the mitochondria. Mitochondria are organelles for energy metabolism in all our cells. They have two membranes, and the particular thing about these structures is although they contain about 1,000 different proteins, they must import most of these proteins from the cytosol where they have been synthesized on cytosolic ribosomes. At the time when I when I just arrived in the lab in 1985, it had become clear through experiments in Neupert's lab and also in the lab of Gottfried Schatz in Basel that when these proteins import into the mitochondria, when they translocate across the membranes, they must be unfolded. And so several things came together. We realized that the mitochondria could be an interesting model to actually ask some fundamental questions about how proteins would then fold in the physiological environment once they translocated across the membranes. How would the folding process then occur? At the time, people assumed it would occur spontaneously. In fact, there were suggestions from the Basel group that the spontaneous folding reaction after the translocation was providing the driving force for the translocation process itself. At the time, we got in contact with Hartl. Walter Neupert was instrumental in mediating for me to get to know Art. Here is a picture from our early interactions. One of the visits that Art made to Munich. We had a little party in our apartment. Art is having a glass of Bavarian beer, and he's also wearing a Bavarian hat. So he rapidly adapted to the environment. Now, at the time, he had already selected the yeast mutant that he explained that was defective in the assembly of the ornithine transcarbamylase, and together we could resolve that a variety of protein assembly and, in fact, folding reactions were affected by this mutant. Let me just illustrate this here again schematically for one protein that we studied. This was, as I said, the combination of the work of Ming Chen, a postdoc in Art's lab, and Joachim Ostermann, a graduate student who worked in the Munich lab. We looked at the transport of a protein called dihydrofolate reductase. This is not a mitochondrial protein. It's a model protein that had been used previously for similar studies of translocation. And we attached a mitochondrial targeting signal, the address label that Art has already mentioned. So this is sufficient to target the protein into the mitochondria. And what we found was that in the mitochondria, this protein did not fold spontaneously, but it first formed a complex with another large protein, which we already knew was HSP60 from the prior work with Ming Chen. And upon addition of ATP to such a complex, we observed that the protein folded into a protease-resistant conformation, very likely representing the native state of this particular protein. This complex could be isolated, and one could actually induce the folding reaction by adding back ATP in an extract of the mitochondria. And this was the case even though the model protein that we used in this particular study, the DHFR, was perfectly capable of refolding spontaneously in the test tube in the Anfinsen type reaction. So we concluded that even proteins that can undergo spontaneous folding in the cell may require factors to assist a folding reaction, to catalyze the folding reaction in an ATP-dependent process. Now, the HSP60, of course, is the homologue of the famous GRO-EL chaperonin that Art and our group then first together and then separately investigated in its functional properties. Now, what I like to do today is to first discuss with you and summarize again why we believe cells require such chaperones for protein folding. After all, that's an important question given the findings by Anfinsen that folding can in principle occur spontaneously. I will then try to retrace the steps a protein has to go through from its synthesis on the ribosome, from the nascent polypeptide up to the folded protein structure by going through different types of chaperone interactions. First, with a set of chaperones that we call nascent chain binding chaperones. These include the HSP70 system, and then transfer and final folding within the GRO-EL cylinder. And then in the last talk, which will be more brief, the last part of the talk will discuss the implications of chaperones in proteotoxicity, in diseases that are associated with protein misfolding and aggregation. Now, after we had seen that mitochondria require such a machinery for folding, we were soon shown by our own experiments and by experiments that many other groups were doing that the mitochondria are not an exception, but in fact all types of cells have these molecular chaperones, have the chaperonins, these cylindrical complexes, and in each case they are essential for the survival of these cells because they are required for the folding at least of a subset of cellular proteins. So why are these chaperones necessary? I'd like to come back to why they are actually called chaperones. And this has really good reasons because in many regards the molecular chaperones are quite comparable to what the human chaperone is doing. You probably have never seen a human chaperone because human chaperones are no longer in fashion. I guess also not in fashion in Hong Kong. But they used to be in the 18th and 19th century. And here you can see a human chaperone in action. So the human chaperone obviously had to prevent illicit or premature interactions between young people when they were not yet married, because this was considered a serious problem, and I think in some societies it still is. But once everything was in order and the marriage contract, so to speak, was signed, people were getting married actually, then the chaperone, of course, would no longer participate in the interaction. Now, incidentally, it has been pointed out to me that this particular couple doesn't really need a chaperone because, as you can see, there isn't a lot of attraction between them. There is rather an aversion. But this may not be the typical situation. Now, in the molecular world, of course, the improper interaction means potentially the clumping together, the aggregation of misfolded or partially folded polypeptide chains. And this needs to be prevented by all types of cells with major efforts. In fact, hundreds of proteins are devoted in cells to control this type of off-pathway reactions. And the reason is that when aggregates form, they can be harmful to cells, they can be toxic to cells. And this is in fact why protein aggregation causes degenerative diseases. Now, when proteins fold, they populate various types of folding intermediates. Art has already alluded to that. There are these energy funnel diagrams. We know that proteins with more complex structures may be trapped in kinetic states, may be kinetically trapped in misfolded states or partially folded states. And these intermediates typically expose hydrophobic amino acid residues and regions of unstructured polypeptide backbone to the solvent. These are the features that will ultimately be buried in the folded structure of the protein, which of course for soluble proteins contains a tightly packed hydrophobic core. But during the folding process, these features may be exposed, and they can give rise to intermolecular associations, intermolecular interactions, and can lead to the formation of aggregates in a highly concentration-dependent manner. Now, these off-pathway reactions need to be blocked, and that's where the chaperones come in. Various families, various classes of molecular chaperones that are structurally unrelated can fulfill the purpose of shielding such hydrophobic surfaces of folding intermediates in such a way that aggregation is blocked, but the forward reaction towards the native state is still allowed and is actually promoted. The chaperones act by not recognizing the sequence of a specific protein, but by recognizing the redundant structural features that characterize non-native states, mostly hydrophobicity. And by doing so, they can interact with many different types of proteins. Many different terracotta soldiers can be prevented from clumping together by these chaperones. But the chaperones are faithfully distinguishing between folded and unfolded or non-native structures. Now, these mechanisms are oftentimes ATP regulated, the binding and releasing of the chaperones, as you just heard for the chaperonins. And they require, in many cases, further cofactors, regulators, so-called co-chaperones. Together, these factors, and this is really now an emerging theme, are forming a chaperone network, a proteostasis network of factors that also connects the chaperone machinery, as you will see later, with the machinery of protein degradation. Now, protein aggregation is strongly favored, we believe, under cellular conditions because aggregation reactions can be enhanced by the macromolecular crowding, the crowdedness that characterizes the cellular solution. Here is an image of the interior of an E. coli cell, a rendition from the work of Adrian Elcock. This is a model where he put all the known protein molecules of an E. coli cytosol in their correct number and concentrations together. You can see that this is an extremely dense environment, perhaps 300 grams per liter of protein, and other macromolecules cause excluded volume effects that would enhance the interactions between non-native protein molecules. And in fact, there is good evidence from in vitro experiments that when you produce similar crowdedness using polymers, you can actually observe now the aggregation of folding proteins, even for those proteins that would otherwise fold spontaneously with good efficiency. So this is probably one reason why chaperones are necessary and why they probably have been necessary very early in evolution for even these types of crowded cells to evolve. Crowdedness, of course, being a prerequisite for life. Now, another important difference between the Anfinsen reaction and the folding in vivo is that when proteins fold in the cell, this process occurs or could in principle begin in the context of the vectorial translation of the polypeptide chain on the ribosome. Proteins are not simply present as a complete polypeptide chain as in the Anfinsen experiment, but they are synthesized step by step on these machines. And this has important implications because the large ribosomal subunit, we know this from the structural work of the ribosome, has a narrow exit channel, approximately 100 angstrom in length, and the polypeptide chain inside this channel is not able to undergo a productive folding reaction. The polypeptide, as long as it is on the ribosome, which is what we call the nascent polypeptide, is topologically restricted. So during this phase of translation, the polypeptide chain, therefore, may only adopt partially folded states. They could still be prone to misfolding, perhaps aggregation reactions. And we believe, therefore, and there is now very strong evidence for many systems, that as the chain translates, chaperones will shield hydrophobic peptide regions that are exposed by these nascent chains. There are different types of chaperones that act at this step. These are the nascent chain binding chaperones. For example, members of a very large family and highly conserved family of molecular chaperones called HSP70. HSP, of course, the abbreviation stands for heat shock protein, because these are also chaperones that are strongly induced by cells under heat stress conditions, but they are also essential under normal growth conditions. Other factors bind directly to the ribosome. I'll come back to that in a moment. For example, trigger factor in the bacterial system. So there are chaperones that essentially wait for the polypeptide chain as it emerges from the ribosome. The HSP70 in particular can bind and release the polypeptide substrate in an ATP-dependent reaction. And this may be useful in allowing the polypeptide to compact, perhaps to form co-translational structure. As we are looking at larger multi-domain proteins, there is strong evidence, particularly for the eukaryotic system, that the modules of a longer polypeptide chain may begin to fold as the chain is being synthesized on the ribosome. So most proteins may interact with this first set of chaperones. And this might be sufficient, particularly for those proteins with simpler structure. But we know that a subset of proteins, perhaps on the order of about 10% or so of cytosolic proteins, require, in addition, the interaction with the chaperonin cylinder, for example, the HSP60 of mitochondria, the GRO-EL in the bacterial cytosol, or the trig CCT in the eukaryotic cytosol, in order to fold efficiently and at a biologically relevant time scale. And this, of course, is a very elegant solution to the problem of protein folding by allowing a single polypeptide chain to fold in a secluded space, in a chamber, where it is encapsulated for a couple of seconds, and therefore unable to aggregate during this time with other polypeptides or macromolecules. Now, these two types of reactions, the types of chaperone principles, have been highly conserved in evolution. They are parts of coherent cellular folding pathways. In each system, in bacteria, archaea, eukarya, you have these upstream components that interact with the nascent polypeptide as it emerges from the ribosome, allows folding of a subset of proteins, and mediates transfer of another subset to the downstream cylindrical chaperonin. In the eukaryotic cytosol, we have added complexities through the existence of parallel chaperone pathways. In particular, there is an HSP90 chaperone system that has been expanded in its significance compared to the bacterial system. It is, for example, required for the folding of many signaling molecules, protein kinases, steroid receptors, and therefore is of considerable medical significance. Again, this system functions downstream of the nascent chain binding chaperones. Let me now look in more detail at the bacterial system. This is the model system that we and others have studied in most detail, and where we have beginning to get good quantitative understanding for the fluxes of proteins through the different types of chaperones, and of course where we have at the same time a good understanding of the mechanisms, of the structures of the participating machinery. In the bacterial system, we have on average relatively small proteins. The average size of a E. coli protein is about 30 kilodalton or so, compared to 50 to 60 kilodalton in eukaryotic cells. And we also have relatively rapid elongation speeds on the ribosome. That means these proteins are synthesized rapidly, together with the fact that they are on average relatively small, makes us believe that this system favors a post-translational folding reaction. This is not to say that larger polypeptides begin to fold on the ribosome, but for the majority of chains, we think the chaperones that interact with the nascent polypeptide function in maintaining the protein in an unfolded but non-aggregated and therefore folding competent state, up to the point where sufficient structural information is then available for folding to go to native-like structures. This usually requires at least the formation of a complete protein domain or folding unit. So most proteins may interact first with trigger factor, which is bound directly to the ribosome, a chaperone next to the polypeptide exit site. This may be sufficient for most small proteins. Longer protein chains interact subsequently with the HSP70 system. This is the DNA-K protein of E. coli and its cooperating factor DNA-J. DNA-J belongs to the HSP40 family. These numbers indicate the sizes of these proteins in kilodalton. So HSP70s usually cooperate with HSP40s, which are regulators of the ATPase of HSP70, but also chaperones in their own right. Now, both trigger factor and the HSP70 system recognize hydrophobic peptide segments that are exposed in a nascent chain or in a non-native polypeptide context. These are the regions that will have to be buried in the interior of the chain when folding is completed. And this, of course, immediately explains then why the chaperone no longer interacts when folding has occurred. These are not consensus sequences, but rather redundant structural features. The trigger factor and the HSP70 system, this has also been shown both by work in Bernd Buchhaus' group in Germany and by Sarah Tita when she was in my lab, have overlapping functions, so there is partial redundancy at the level of these upstream factors, whereas the GROE-L-GROE-S system, about which you just heard, is always essential. It's in fact the only chaperone, to my knowledge, in the bacterial system that is always essential under all tested growth conditions, suggesting, of course, that there must be a set of proteins that strongly depend on this particular folding environment to go to native structures. Now, the HSP70 system plays a major role. It is thought to be a central hub in the chaperone network. And more recently, we have undertaken a quantitative analysis to understand what are the types of proteins, how many different polypeptides interact with HSP70. And in particular, we wanted to know in more detail how this chaperone system cooperates with the downstream chaperonin in a folding pathway. Such a folding pathway had been suggested many years earlier by Thomas Langer when he was in my lab, based on in vitro reconstitution experiments. And in a few moments, I will show you such an experiment. But let me first explain what we found when we carried out quantitative proteomics with pull-down reactions from cells in which we isolated HSP70 with its bound polypeptide spectrum. We saw that HSP70, the DNA K, interacts with approximately 700 different newly synthesized polypeptides. And when the trigger factor chaperone was deleted in this strain background, then the number of interactors of the HSP70 increased by an additional 350 protein, reflecting the partial redundancy of these two systems. In fact, the trigger factor absence resulted in the increased interaction of small proteins with the HSP70 system, proteins that would otherwise have preferentially interacted with the trigger factor. So a large fraction, on the order of 30% or so, of newly synthesized polypeptide chains interact with these upstream factors. And this is probably an underestimate because these analyses, of course, are never complete. The HSP70 system was found to interact predominantly with proteins that are larger than average and have structural features that render them aggregation prone, increased numbers of hydrophobic amino acid residues or certain structural properties. There are now excellent prediction programs that allow one to classify whole proteomes according to the aggregation profile. Now, what was very interesting and in fact rewarding to see was that there was a huge overlap between the substrate interactome of HSP70 and that of the GROE-L chaperonin, which we had previously characterized and found to interact with approximately 250 or so different cytosolic proteins. We found that the previously identified GROE-L substrates occupy approximately 30% of the DNA K capacity. So whatever is folded by GROE-L and utilizes the GROE-L system is strongly regulated by the interaction with the upstream chaperones. So there is a cooperation of these two systems in a chaperone pathway that is very relevant in a quantitative context. The obligate GROE-L substrates, about which I will talk in a few moments, accumulated further on the DNA K. When GROE-L was depleted, we could not delete the GROE-L because it's an essential component, but we can use a strain background in which we can deplete the cells of GROE-L. Under these conditions, then the DNA K functions as a buffer for the GROE-L dependent proteins, transiently preventing their aggregation. And many of these proteins will then be guided either to the degradation system, and only when the system is exhausted do we observe aggregation of these proteins. This has recently been published. Now, the pathway of cooperation of 70 and GROE-L was proposed many years earlier by Thomas Langer's experiments. These were pure in vitro reconstitution experiments. The reason why we carried them out was we wanted to know why cells actually have these two different types of chaperones, the chaperonin GROEL on the one hand and the HSP70 system, even though it appeared that both systems serve the same purpose. Thomas used a protein called rhodanese, which is a mitochondrial model protein, and we used it in the refolding studies because it was known to have a very strong tendency to aggregate. When he diluted this protein from denaturant into a buffer solution containing only the HSP70 system, he did not observe refolding of rhodanase. But what he did observe was a strong stabilization of the protein against aggregation. The protein remained folding competent. And upon subsequent addition of the GROEL system together with its cofactor GROES, efficient transfer from the HSP70 to GROEL occurred, and the protein folded to its native enzymatically active state. This is exactly the type of reaction that seems to occur for a much larger number of proteins in the E. coli cytosol, and we have in the meantime reproduced these reactions for many of the proteins that are now known to be obligate GROEL substrates in the bacterium. The reason why the HSP70 system cannot fold these proteins is intriguing and is still a subject of investigation, but the major difference, of course, is that HSP70 is a monomeric chaperone. It binds to a specific peptide sequence in the rhodanase molecule. It binds and releases the protein into the bulk solution, whereas only the GROEL system forms this chamber in which a folding reaction can occur while the protein is completely sequestrated from the bulk solution. So this clearly is the fundamental difference between these two systems, and proteins like rhodanase require this particular folding environment. Now, how does the GROEL system work? After the initial discovery with Hartl using the mitochondria, we realized that it would be better to now study the bacterial GROEL, the HSP60 homologue, because it was much easier to be prepared in large quantities for in vitro mechanistic studies. These are early electron micrographs that we obtained with Wolfgang Baumeister, my collaborator at the Max Planck Institute. Art got very similar images with a collaborator at Brookhaven, which he mentioned before. And what you can see here is the cylindrical double ring structure, of course at much lower resolution than what was then seen 2 years later in the crystal structure that Art and Paul Ziegler solved. But what was important for us at this stage was to observe that when unfolded protein, in this case the rhodanase molecule, was bound to GROEL, we could observe an additional stain excluding mass, negative stain excluding mass, in the center of the GROEL ring. And this, of course, began to shape our thinking in terms of folding reactions perhaps occurring, at least in part, while the protein was associated with this particular ring system, a problem that kept us then busy for several years until the current model for chaperone action could be established. The next step was to look at the binding of GROES. We could observe that GROES binds on top of one of the two rings in an asymmetrical fashion, apparently resulting in major conformational changes in the interacting subunits. But at this point, we were not yet able to properly interpret these changes. It took 2 more years, and the work of Helen Sabel, Art already alluded to it. She had electron micrographs with higher resolution, which showed that when GROES bound, this created an enlarged volume, an enlarged chamber under the lid of the GROES, which perhaps represented the space in which folding reactions could occur. At about the same time, Jörg Martin in my lab observed that the GROES was not bound statically to the GROEL, but in fact that it carried out an ATP-dependent binding and release cycle, and we had some circumstantial evidence that it was possible for the GRU-ES and the substrate protein to be bound to the same ring of GRU-EL. Together, these findings suggested and made it much stronger for us to believe that, in fact, the folding reaction could occur inside the central chamber. And here, just to remind you again, is the current model from the work of Art and George Lorimer and other groups and our own work that explains how the GRU-ES system functions. We now believe that polypeptides are delivered probably by the HSP70 system, perhaps other upstream chaperones, in a partially folded state. They are unable, however, to use these chaperones for productive folding. They are transferred into the open ring of GRU-EL, bind via multiple hydrophobic interactions with the apical domains of the GRU-EL ring. And then in an ATP and GRU-ES dependent step, encapsulation of the polypeptide chain occurs. There are major conformational changes in the GRU-EL interacting subunits, whereby the hydrophobic surfaces that are used for capturing the sticky, partially folded protein will be buried, and the inner ring surface now becomes largely hydrophilic and net negatively charged. At the same time, the cavity size is enlarged, and the polypeptide is now free to fold inside this chamber in a reaction that is timed by the ATPase of GRU-EL. It takes approximately 10 seconds or so, after which time then binding of ATP to the opposite ring sends a signal that allows the ring, the cis complex, to dissociate. The GRU-ES opens up, and native protein can emerge if the folding reaction was successful in a single round. If not, a partially folded protein would be recaptured and can undergo another round and another round of attempted folding inside this cage until it is eventually either successful or perhaps becomes transferred to the degradation system. The two rings act sequentially. Somewhere here, another protein could bind in order to be encapsulated and undergo folding, but this is not shown here for simplicity. Now, what are the proteins that require this particular system? We knew from earlier work in our lab and in Art's lab that it's not the majority of E. coli proteins, but only a small fraction that require GRU-EL. We carried out, again, pull-down reactions to identify the interactome. This was work in collaboration with Matthias Mann's group, who is now in our institute, but at the time he was still in Denmark. At this point, we expressed in E. coli cells a version of GRU-ES with histidine tags attached, so that we could then isolate easily these GRU-EL complexes containing the substrate. This was achieved by rapidly converting ATP to ADP during cell lysis, stabilizing the GRU-ES complex in a non-cycling state. And what we then can observe is in such pull-downs that here we have the GRU-EL subunit, this is the GRU-ES with his tags, and in between you see this smear of polypeptide bands. These are essentially the different polypeptides that utilize the GRU-EL system, apparently undergoing folding with the GRU-ES machinery. We could quantitate these proteins, we could identify them by mass spec, consistently finding 250 different specific substrates. It was then the work of Michael Kerner mostly, who classified these proteins according to their dependency on GRU-EL, both through in vitro studies and through in vivo experiments. And he suggested that there are approximately 80 different proteins that we can identify that we call class 3 proteins, meaning that these are proteins that are obligatory dependent on GRU-EL for folding, that they cannot use any other chaperones for folding to the native state. These proteins include a number of essential proteins. They occupy the majority of the chaperone capacity, and structurally they are complex, have preferentially alpha beta and alpha plus beta domain topologies. These are protein domain topologies with many long-range interactions, and we think that these are proteins that have a particularly strong tendency to form these kinetically trapped states, which are then aggregation prone and disrupt the efficiency of folding in vivo. Now, most of these proteins, these class 3 obligate substrates, are smaller than approximately 50 kilodalton, which means that they fit nicely into the chamber of the GROEL-GROES system. We find relatively few proteins that are larger in size. We are not quite sure yet how they utilize the GROEL system, but Hartl's work has proposed that some of these proteins may bind and release in a manner without encapsulation by GROES, whereby they bind and release from the trans ring, and this could also increase their folding efficiency. The fate of such proteins could be tested in vivo by depleting the GROEL using a strain where the GROES and GROEL are under arabinose control, and then we shift the cells to glucose. Within a few hours, the GROEL will be depleted as the cells continue to grow and then eventually die after 7 or 8 hours. You can see that proteins that we classified as class 1, which has very little GROEL dependence, at least in vitro, were rather unaffected. The enolase is a very abundant protein, and we find this protein only interacting with GROEL because it is so abundant. Proteins like TDH, which are belonging to class 2 and which are highly chaperone dependent, however, can use either the GROEL system or the HSP70 for folding. Again, these proteins are unaffected when we deplete GROEL, but an obligate class 3 protein such as METK or DAPA or other proteins of this category will be depleted from the soluble fraction of the cell, and in this case appear in the pellet fraction containing protein aggregates, insoluble protein, or these proteins, another fate that we frequently observe, will be degraded, will essentially disappear from the cell as they are degraded upon synthesis and being unable to fold. Now, these GROEL obligate substrates oftentimes have one particular domain structure, namely the Tim-Bevel structure, which is characterized by hydrophobic beta sheets in the center of the molecule surrounded by amphiphilic helices, a result that was also recently confirmed by the group of Taguchi in Japan. So it's the predominant fold that GROEL has to interact with. And we know from in vitro studies that these proteins oftentimes have complex folding pathways, presumably with rugged energy landscapes. Now, an important question, of course, is how does the GROEL achieve the efficient folding of such molecules that fail to fold with the upstream chaperones? And the question that comes up is whether GROEL modulates the energy landscape of folding, and Hartl has already alluded to this question. This is to some degree an open question still, and that needs to be resolved by further experimentation. But we would suggest that in addition to preventing aggregation during folding, the confining environment of the GROEL-GROES cage may also contribute to smoothing the energy landscape of such folding of such proteins, and thereby perhaps accelerate the folding of certain proteins to a level where folding is then allowed to occur at a biologically relevant time scale. Questions such as the physical properties of the wall of the cage play a role here, perhaps effects of ordered water structure oriented on the negatively charged inner wall of the cavity. These are questions that remain to be addressed. Now, to summarize this part of the talk, we are now beginning to get good understanding for the chaperone network in the E. coli cytosol, and indeed can use this model to understand more complex chaperone networks in the eukaryotic system. When proteins are synthesized on ribosomes, the vast majority of them, at least for the cytosolic proteins, seem to interact first with trigger factor. And many proteins, approximately 700 at least, will then be transferred to the DNA-K, the HSP70, as a subsequent step of chaperone interaction. This would be proteins that are somewhat larger and have more complex structures. There is partial redundancy between trigger factor and DNA-K. If we delete trigger factor, many of these proteins can reach DNA-K directly. However, trigger factor retains some specific functions that have only recently been established, also through the work of Jonathan Weissman. For example, in the case of the folding and assembly of certain beta-barrel proteins of the outer membrane, and also ribosomal and small basic proteins. HSP70 system, apart from mediating folding of proteins through ADP-dependent cycling, also has a major role in transferring substrates and selecting substrates to GRO-EL. It's a little bit like a distillation machine. You have a chaperone that acts upstream. Those proteins that can use this chaperone for folding through successful binding and release cycles will no longer interact with GRO-EL. Those proteins that get kind of stuck to the HSP70 because they are unable to fold will then eventually be transferred to GRO-EL to reach their final states inside the GRO-EL chamber. Now let me briefly discuss that what happens when proteins misfold and aggregate. Proteins do not always fold with very high efficiency despite the existence of molecular chaperones. I would like to stress that molecular chaperones are an important and integral part of a much larger network of factors. They are required, of course, for multiple functions during de novo folding, prevention of aggregation, refolding, remodeling of proteins during stress, and so on. But they also cooperate, and this is very important, with the ubiquitin proteasome system for proteolytic degradation of terminally misfolded proteins, and also with the system of autophagy and lysosomal protein degradation. That is a way to degrade and remove certain types of protein aggregates that could be dangerous and play a role in disease. But despite these organized pathways, which we do not yet fully understand, protein aggregation does occur and is associated, as you know, with an increasing number of neurodegenerative diseases in particular, such as Alzheimer, Huntington, amyotrophic lateral sclerosis, Parkinson's disease, the prion diseases. All these diseases have in common the formation of certain types of aggregates that are called amyloid and have fibrillar structure, cross beta structure, where beta strands run perpendicular to the long axis of these fibrils. Now, in many of these cases, the aggregation phenomena only occur later in life, and it has been suggested that the manifestation of these diseases may be caused, or at least supported, by a decline in the available chaperone capacity that may occur during aging. So this is summarized here in this schematic model, that manifestation of some of these diseases at least appears to be a result of an imbalance between the production of misfolded proteins that seems to occur all the time and the available cellular capacity of chaperones to deal with these potentially dangerous molecules. A classic example for this type of disease is Huntington's disease and a group of other diseases that are caused by proteins which, due to heritable mutation, have an expanded polyglutamine tract in their sequences. In the case of Huntington, we know that people that do not get the disease have between 9 and 36 or so glutamines in the amino terminal exon of the protein, whereas people with 38 or more than 38, up to 100 or so, will get the disease at some point in life, whereby the length of this polyQ segment correlates inversely with the time of disease onset. The longer the polyQ segment, the earlier you get the disease, and the stronger the aggregation propensity of the protein is, as could be established in vitro studies. So this is actually a wonderful model for testing whether chaperones are indeed capable of preventing such toxic protein aggregation phenomena. And we can do so in an in vitro setup. This is an experiment that Paul Mukowski carried out when he was a postdoc in the lab in 2000. And what you can see here is an experiment with a control protein having 20Q. This is in the normal range. It does not form detectable aggregates in a filter retardation assay, whereas the protein with 53Q forms time-dependent fibrillar aggregates. These are highly stable SDS-resistant structures. And when we add components of the HSP70 system in mild stoichiometric excess, HSP40, HSP70, you can see that the formation of these aggregates is strongly retarded. And interestingly, for those protein molecules that still aggregate, we do not observe the formation of fibrils, but we observe the formation of amorphous aggregates. So we concluded from these early studies that if sufficient chaperone capacity were available in these cells, aggregation of the Huntington disease protein could be prevented. And in fact, overexpression of chaperones could be demonstrated to suppress the toxicity of mutant Huntington. Apparently, the chaperones are capable of deviating the protein from a potentially toxic aggregation pathway that leads to the fibrils as the endpoint to a less toxic or benign aggregation pathway. And as we increase the chaperone capacity more and more, we can completely prevent aggregation and presumably direct the protein more successfully towards degradation. Now, the next question that we asked, and which I want to end with, can we use the power of chaperones perhaps by activating the system pharmacologically and developing therapies to treat these diseases in the future? The answer is at least yes, in principle. We could use here a model drug, small molecule geldanomycin, which was known to induce the stress response by inhibiting a particular chaperone, the HSP90 that I already mentioned, and thereby activating the transcription factor that is responsible for the expression of various types of chaperones and stress proteins, including HSP70, HSP40, and HSP90 itself. As you can see, when monkey cos cells are treated with increasing but low concentrations of this inhibitor, you will see that the concentration of these chaperones increases perhaps 2- or 3-fold. This is completely sufficient to then prevent the aggregation of a Huntington construct with 72 glutamines, well in the pathological range, that was expressed as a fusion with GFP, so that you can nicely see how this prevention of aggregation actually works. This would be the control condition where we have large perinuclear aggregates of Huntington. And as we increase the concentration of this drug, we will see that the aggregation of Huntington is now completely prevented. The protein is diffusely distributed, fills out the whole cell. And with these types of systems, it has been shown also that you can, in fact, suppress toxic effects of these molecules. The downside of components such as geldanomycin, of course, is that they inhibit HSP90, which is in fact an essential chaperone. So you probably have significant side effects. So this will probably not be the molecule that could be used as a real treatment. It just demonstrates the principle that one can search for compounds that have such effects, and eventually, hopefully, we will find some that are less toxic. Now, one important question, of course, in this whole field is why is actually the aggregation reaction of such proteins toxic? This is not at all clear from first principles. What we do understand is that a protein that aggregates will not be functionally active, but this is not the problem that is the primary aspect of these disease molecules. What actually happens is that when these proteins aggregate, they acquire a new property, a gain of toxic function, which seems to be largely independent of their normal biological role. Probably there are multiple pathways of toxicity, and the way in which the HSP70 system interferes with this toxic cascade is to act very early on, as we found with fluorescent labeled molecules using FRET experiments, probably by preventing the intramolecular conformational change that occurs in the expanded polyQ sequence, and that then acts as a nucleation phenomenon, a nucleation step for the formation of first oligomers and then larger amyloid fibrils. We also found, interestingly, that the HSP70 can cooperate in this reaction with the cytosolic chaperonin, and this supports a process in which the protein is deviated from the toxic pathway of aggregation to disordered aggregates, reminiscent of the cooperation between these two types of chaperones, the cylindrical chaperonin and HSP70, in the de novo folding of many proteins, as I have shown you. Toxicity may arise from interactions with membrane structures, from interactions with other proteins, thereby interfering with the function of multiple key molecules. Evidence for multiple mechanisms of toxicity has been presented, and certainly much more research is necessary to understand this. Now, in the future, we hope that perhaps chaperones can help us to come closer to the fountain of youth that is shown here in this nice Renaissance painting from Lucas Cranach. Maybe not keeping us younger forever, but helping us to delay some of the diseases of aging, some of these dreadful neurodegenerative diseases in particular. That would, of course, be wonderful. What you can definitely see here is that people in the fountain of youth would have a need for chaperones because they are all naked and swimming around here very happily and doing all kinds of things that are normally prevented by molecular chaperones, human chaperones. So let me end on this note and show you the names of people. I have had, of course, like Art, many collaborators and wonderful co-workers, many talented students and postdocs. I have mentioned the names of those who performed the critical experiments, but I want to stress in particular the long-term collaboration and the important contributions that Manojit Hartl, who is also here in the audience, has made to all our work. Thank you very much.
Thank you very much, Professor Hartl, for the enlightening talk. We have time for a few questions.
We know in nature there's a lot of here. Okay. We know in nature there's a lot of protein contain in intrinsic disorder regions.
Yes.
And my question is how chaperone differentiate this protein from those really unfolded protein?
That is actually a very interesting question, and I'm not sure that I can really give you a final answer on that. It hasn't been systematically studied whether such proteins are substrates of chaperones or to what extent they are substrates of the chaperone system. What we do observe, however, is that these proteins that preferentially populate unstructured conformations oftentimes are more highly charged than average proteins. They lack a tightly folded hydrophobic core. In principle, one would assume that these are not the molecules that are preferentially recognized by molecular chaperones. And perhaps this is one reason why some of these molecules, at least, belong to the most dangerous proteins, apparently, in the cell when they do undergo aggregation. They don't seem to have a very strong tendency to aggregate on their own, but when it happens, they seem to be doing so in a probably relatively uncontrolled fashion and can then give rise to the formation of toxic aggregates, for example, alpha-synuclein in the case of Parkinson's disease. It is also not clear to what extent these proteins exist in their free form as unstructured polypeptides in the cell, and to what extent are they mostly in perhaps transient interactions with multiple interaction partners. Oftentimes it is shown that such molecules occupy important control positions in protein networks, hub positions, and have many interaction partners. What we found recently was that proteins that undergo fibrillar aggregation and cause toxicity apparently have a tendency to interact promiscuously with other cellular proteins that are enriched in unstructured regions. So I believe that unstructured proteins play a very important role in understanding these mechanisms and the functioning of the proteostasis network and how it can lose its control, but it is not yet fully understood how they actually interact with the chaperone system. Yes?
I also asked that how does the polypeptide chain folds in the chaperone, cylindrical chaperone?
In the which chaperone? In the
cylindrical chaperone.
In the cylindrical chaperone.
Yeah, is it the same way? Does a polypeptide chain folds
and that
in?
is absolutely one of the last remaining questions regarding this system. And while it is clear that certain proteins, particularly those that are perhaps not occupying the central volume of this cage fully, dihydrofolate reductase, in fact, as Hartl has demonstrated, seem to fold, as far as we can tell, by the same mechanism as they would in free solution. We have suggested that perhaps larger proteins may, by some interaction with the cavity wall, be engaging different elements or different aspects of the energy landscape more preferentially, perhaps whereby the confinement effects that I suggested would destabilize certain types of intermediates. We believe that many of these molecules that are chaperone independent have long search times. They need long search times to find native interactions within the molecule. Such a reaction could potentially benefit from an element of steric confinement, coupled perhaps with other physical effects that may be exerted by the cavity. But as I said, this is still an issue that requires a lot further work.
May I ask why the chaperone availability will decrease during aging?
Yeah, you may.
Well, that's what I was just
But I will not be able to answer that question really. Of course, there are some fundamental considerations when you look at model organisms such as the worm C. elegans, for which such findings have been predominantly made, and also I think in Drosophila. The work particularly I'm referring to is from the group of Richard Morimoto's laboratory. It is conceivable that evolution has optimized the whole network of chaperones and the reactions of protein folding and proteostasis only up to the point where an organism has been able to propagate. In the case of the worms, after their fertile phase has ended, it appears that a decline in proteostasis occurs, whereby we don't know exactly whether the concentration of chaperones declines or whether the chaperones are no longer in a functional state. Perhaps they undergo modifications. What is clear is that the system is no longer able, in particular, to react to various forms of stress that would normally require an upregulation of the chaperone system. For example, the heat stress response becomes much less potent as these animals age. So these are factors that will then contribute probably to an increased propensity of molecules to form misfolded states and eventually aggregate. We have recently undertaken a large-scale quantitative proteomic study together with the Mann group in the worm system, and it is very clear that, let's say from day 5, 6 or so on of adulthood, we begin to see accumulation of protein aggregates, and we see changes in the abundance of various elements of the proteostasis system, including chaperones and also components of the proteolytic system that could favor the formation of misfolded proteins. Why this really happens, of course, is not clear, and to what extent this can be transferred to mammalian cells is still a completely open question. I personally do not believe that these findings can be transferred to human cells, for example, to the same extent as we find these things in the worm.
All right, I think in the interest of the time, maybe we should conclude our today's lectures. Let's thank both Professor Hartl and Professor Howard for the wonderful talk. And before I close the session, I wanted to remind the life science faculty, students, postdocs, that we will have a face-to-face meeting with 2 of the laureates. And in room 5513, we will be providing snacks and drinks. And let's thank 2 laureates again. And thank you for coming.